Now, just before we talk about the chemical reactions involved in photosynthesis, it's important to remind ourselves of the balanced chemical equations for both cellular aerobic respiration at the top, as well as photosynthesis. Now, we know photosynthesis makes glucose. It's the set of reactions that results in the end product, glucose. But we know glucose is needed by all cells of the plant for respiration. So we know cellular respiration is going to break down the glucose that's made from photosynthesis. And that chemical energy is incorporated into many molecules of ATP, which are needed for cells to operate and survive. So I'll pause the video and then, you can pause the video, and then if you just jot down the chemical equations, we'll go through the answers very shortly. So the first equation at the top then for aerobic respiration, well, we know glucose is a monosaccharide, it's a hexose sugar, and we know it's needed by all the cells of the plant. Now we know in the presence of oxygen, we know that's the final electron acceptor. So the final electron acceptor inside the mitochondrion at the electron transport chain. And that pulls high energy electrons through the ETC. They lose energy as they do so, and that energy is used to build the electrochemical gradient that is needed to make ATP. Now we know some ATP is made by substrate level phosphorylation, 4 molecules, and we know the other 34 is made from oxidative phosphorylation inside the mitochondrion. Now we know carbon dioxide is a waste product of aerobic respiration. And we also know water is a waste product of aerobic respiration. And it's the oxygen, the molecular oxygen, that combines with electrons and protons that will form the water at the end of the ETC. Now this carbon dioxide is actually very often the starting material for photosynthesis. So a lot of the carbon dioxide produced from aerobic respiration in the palisade and the spongy mesophyll cells of the leaves is going to be utilized as a starting material for the Calvin cycle. Now we know photosynthesis uses carbon dioxide. The carbon here is the carbon that ends up in the glucose molecule. We also know water is required, and photolysis is the enzymatic splitting of water in the presence of light to produce molecular oxygen, which is a waste product of photosynthesis. And it also provides protons and electrons, and it's the electrons that replace the electrons lost from chlorophyll A in photosystem 2 at the reaction center. So the glucose itself is made in the stroma of the chloroplast. Now, the series of chemical reactions that make the glucose in the stroma of a chloroplast were first elucidated by a scientist called Melvin Calvin. And he basically worked out the organic molecules within this cycle. And he also noted that 2 molecules that were produced in the light from the light dependent stage were needed to drive the Calvin cycle. And these were ATP and reduced NADP. So these are 2 products of the light dependent stage that will be needed to drive the light independent stage that will make glucose in the Calvin cycle. So the cycle itself, there's 2 starting substrates, and the first one is a 5-carbon molecule called ribulose bisphosphate. Now ribulose means 5, so we can see there's 5 carbons in this sugar, and bis means 2, so there's 2 phosphate groups as indicated by the yellow circles. Now ribulose bisphosphate will bind or successfully collide with the active site of an enzyme called rubisco. But the second substrate is carbon dioxide, and that also successfully collides with a slightly different area of the same active site. So we end up with rubisco with carbon dioxide and ribulose bisphosphate successfully collided with the active site. That forms the enzyme substrate complex, and rubisco catalyzes the addition of the carbon from carbon dioxide onto the 5-carbon molecule to form an unstable 6-carbon molecule. Now, because this is unstable, in the stroma of the chloroplast, this splits and it produces 2 3-carbon molecules, each of which is phosphorylated. So this is called glycerate 3-phosphate. So we can see each 3-carbon molecule has a single phosphate group on it. Now, these reactions up to this point, so if we just split the cycle in half, so we draw a line here. The fixation of atmospheric carbon dioxide onto ribulose bisphosphate by rubisco to form the unstable 6-carbon compound, which splits into 2-glycerate 3-phosphate. This can occur in the dark and the light. It does not require ATP or reduced NADP. So it does not require these from the light dependent stage. So even in the dark, rubisco can fix CO2 with ribulose bisphosphate to form 2 molecules of G3P. Now, rubisco is probably the most abundant enzyme on the planet, and we know it preferentially binds or successfully collides with carbon dioxide into its active site with ribulose bisphosphate. It can also successfully collide with oxygen, which fits into the active site. And if the concentration of oxygen is much higher than CO2, then what we end up with is a process called photorespiration. So the levels of CO2 must be high enough to promote the fixation of CO2 over the fixation of oxygen by rubisco. Now, once G3P has been produced, 2 molecules from the light dependent stage, ATP and reduced NADP, are needed to drive the reduction of G3P, or glycerate 3-phosphate, to triose phosphate. So we can see this on the picture. The hydrogen from reduced NADP is used to reduce G3P, and that will reoxidize the NADP, which can then go back to non-cyclic photophosphorylation at the thylakoid membrane to regenerate more reduced NADP. So the main difference here is that it's not NAD as we would find in respiration, it's actually NADP, which is quite important. Now, the energy for this reduction reaction comes from the hydrolysis of ATP, and that will be hydrolyzed by part of the enzyme called ATPase that releases ADP and inorganic phosphate. So because the glycerate 3-phosphate already has a phosphate group attached, as does triose phosphate, the phosphate group from the ATP is not needed. It doesn't need to be added to G3P. It's just the energy released from the hydrolysis of that high energy bond drives the reduction reaction. Now, once triose phosphate has been formed, we know that's the carbon backbone that will be used to make organic molecules like glucose or sucrose, amino acids that we know contain a bit of nitrogen, many lipids, different types of lipids like triglycerides made from fatty acids and glycerol. Chlorophyll is an organic molecule. It's the pigment that absorbs certain wavelengths of light during the light dependent stage, chlorophyll A, and also chlorophyll B, which is the accessory pigment, or one of them. We know proteins are organic macromolecules made from amino acid subunits, and we know things like maltose and sucrose in particular are the sugars that get translocated up and down the phloem. Now, the triose phosphate can also be used to regenerate the ribulose bisphosphate as the starting material for rubisco. Now that will require some ATP, the energy released from the hydrolysis of ATP, to drive the conversion of triose phosphate back to ribulose bisphosphate, the regeneration of ribulose bisphosphate. Now, we have a quick look at a past exam question just in terms of the Calvin cycle. The first question says, name compounds 1, 2 and 3 shown on the diagram. So if you wish to pause the video and have a quick go at these, I'll go through the answers very quickly. Now, if we have a look at molecule number 1 at the top, well, if we count the number of carbons, we can actually see 1, 2, 3, 4, 5 carbon atoms there, the slightly darker circles. And if we know it's got 5 carbons, we know this is going to be ribulose bisphosphate. So that's one of the 2 substrates needed for the first chemical reaction catalyzed by Rubisco. Now we know carbon dioxide is the second substrate, and that's fixed onto the ribulose bisphosphate by this Rubisco enzyme. So we could put Rubisco here. Now the next molecule then, if we have a look further down, well, we know this here and this molecule here, they look the same. And if we count the carbons, those dark circles, we can see 1, 2, 3 carbons here. And again, we've got 3 carbons on the left-hand side over here. So we know the 6 carbon molecule, which is very unstable, formed from CO2 fixation, splits in the stroma of the chloroplast to make 2 molecules of glycerate 3-phosphate. Now we know glycerate 3-phosphate is converted to triose phosphate, so the next molecule in the cycle must be triose phosphate. Now if we look at X and Y, we can start to work out what these must be. Now we said earlier the regeneration of ribulose bisphosphate, so this kind of sequence here, requires ATP. And we know ADP is going to be released because one of the phosphate groups is needed to form the bisphosphate of ribulose bisphosphate, as well as the energy released from this chemical reaction. So X must be ATP. So again, we can put ATP up here and ADP, in this case, plus inorganic phosphate, because the phosphate group is not needed because G3P is already phosphorylated, as is triose phosphate. So that must mean that Y down at the bottom is reduced NADP, and it's the hydrogen from reduced NADP that is used in the reduction reaction from G3P to triose phosphate. So we know that's going to release NADP, reoxidized NADP. So if we have a quick look at the answers then, number 1 was ribulose bisphosphate, the 5 carbon starting substrate for Rubisco, along with carbon dioxide. Number 2 was glycerate 3-phosphate, so that's formed from the unstable 6 carbon molecule in the stroma. And we know number 3 is triose phosphate, otherwise known as glyceraldehyde 3-phosphate. So you can use either of those terms. We normally stick with triose phosphate here. Now, when you get a question about an enzyme, it's very important to always reference that the enzyme catalyzes a specific reaction. So one type of specific reaction. So Rubisco is ribulose bisphosphate carboxylase oxygenase, and we know it fixes CO2 by catalyzing this reaction with ribulose bisphosphate. So please always remember to use the word catalyzes in your answers. Right, we know X, like we said, is ATP. We know every time ATP is hydrolyzed, the high energy bond at the end of the molecule, 30.6 kilojoules per mole of energy is released, which is used for a certain type of process. Now, Y is reduced NADPH2, like we said. Now, we know the difference between this and respiration is that it's NADP, so it's actually got a phosphate group on it, as opposed to just NAD. Now, if we start to look at A, B, and C and what's happening during these processes, well, we know A at the top. If we just highlight this process here in the Calvin cycle, this portion is carbon dioxide fixation. which is listed here on the right hand side. Now we know B, because triose phosphate is being used to remake ribulose bisphosphate, it must be the regeneration of that 5-carbon molecule. And lastly, C, we know because G3P is reduced to triose phosphate, this must be a reduction reaction because it's referencing this step here, reduced NADP, giving off its hydrogen to be able to reduce the G3P to triose phosphate, and the NADP becomes reoxidized, otherwise known as a redox reaction, because reduction and oxidation occur side by side. Now, this is a very typical exam question that you might see on an exam script. Now, the graph represents the amount of radioactive carbon incorporated into 3 molecules: glycerate 3-phosphate, glucose, and ribulose bisphosphate. Now, the radioactive carbon is C14. So what they've done is they would have put a bag probably over a leaf, and inside that bag would have been radioactive carbon dioxide. Now we know the carbon dioxide is fixed due to the catalytic activity of ribisco with ribulose bisphosphate in the stroma of the chloroplast. So any radioactive carbon will ultimately be incorporated not only into glucose, because that's made from triose phosphate, but also back into ribulose bisphosphate, because some of the triose phosphate made from CO2 fixation is then used to regenerate the ribulose bisphosphate. So G3P at the top, glucose, and ribulose bisphosphate will all ultimately have radioactive carbon in them from the radioactive carbon dioxide. So if we look at the scenario in the light then, so what's happening during this stage? Well, the dotted line for glycerate 3-phosphate, so if we look at this very carefully, we can see it goes up. Now that's because the carbon dioxide with the radioactive carbon is being fixed with ribulose bisphosphate, and that forms an unstable 6-carbon compound. And because it's unstable, it splits into 2 molecules of G3P. So as the Calvin cycle keeps going round these series of reactions, more and more radioactive carbon is getting fixed, and more radioactive carbon becomes incorporated into G3P. Now it's the same for ribulose bisphosphate. Because G3P is converted to triose phosphate with the radioactive carbon, the triose phosphate is then used to regenerate ribulose bisphosphate, which will also incorporate the radioactive carbon. So the level of ribulose bisphosphate with radioactive carbon also increases. Now eventually all the carbon in ribulose bisphosphate will contain radioactive carbon. So that's why the relative amounts of radioactivity remains constant in the ribulose bisphosphate, because all the 5 carbons in each of these RUBP, ribulose bisphosphate molecules, is radioactive carbon. Now the relative levels of G3P compared to ribulose bisphosphate, there's about double the amount of G3P, or certainly a lot higher amount compared to ribulose bisphosphate, and that's because we know ribulose bisphosphate is fixed with carbon dioxide, and we know that unstable compound, the 6-carbon, splits into 2 G3P. So for each ribulose bisphosphate, we end up with 2 glycerate 3-phosphate, which is why there's really nearly twice as much G3P compared to ribulose bisphosphate. Now, we would think ribulose bisphosphate, because it's being used in CO2 fixation, that the levels would decrease in the light. However, because G3P is being made, we know G3P in the presence of ATP and reduced NADP, so ATP and reduced NADP from the light dependent stage, they're available. The the G3P is being converted to triose phosphate, and some of the triose phosphate is being used to regenerate ribulose bisphosphate. So even though ribulose bisphosphate is actually being used up, so it is being used up, it's also being regenerated from triose phosphate, and that's why it stays constant in the light. Now, we know in the light the triose phosphate is used to generate glucose. So we see over time, which is the x-axis along the bottom, so we could put x-axis here. Over time, the glucose being made starts to increase in the light because more and more triose phosphate is being made, therefore more and more glucose is being made. So the glucose, we can see here, the dotted line goes up in the light. However, when we switch the light off, what we'll see is the level of ribulose bisphosphate decreases. And that's because we know in the dark, when there's no light, Rubisco can still catalyze the fixation of CO2 to ribulose bisphosphate. So the ribulose bisphosphate is being used up to make the unstable 6-carbon molecule. Now, we know the unstable 6-carbon molecule will split, and when it splits, we know it makes G3P. So the relative level of G3P goes up because that's still being synthesized. However, because there's a lack of ATP and a lack of reduced NADP in the dark, because the light dependent stage is not taking place, so we could say the light dependent stage does not occur here. There's no ATP, there's no reduced NADP. So G3P is not being reduced to triose phosphate. And if there's no triose phosphate, there's no more glucose being synthesized. So that stops going up. And also there's no more ribulose bisphosphate being regenerated because of the lack of ATP. So because ribulose bisphosphate is being used up, it goes down. It's not being replenished. G3P is still being made from the ribulose bisphosphate that fixes onto CO2, but it's not being used to generate triose phosphate, so it goes up. Now, the glucose, you can see the dotted line, does actually go down somewhat. So we can put this on the graph here. So even though triose phosphate is not being formed, we might expect the level of glucose to stay constant in the dark because no more glucose is being synthesized from the triose phosphate. But it actually starts to decrease somewhat in the dark because we know glucose is also being used up in respiration. So the glucose that is present will continuously be used in cellular aerobic respiration, and so the levels of glucose will gradually start to decline because no more glucose is being produced in the dark. Okay, one more exam question that we could have a look at about the Calvin cycle in the stroma of a chloroplast. Now the first question says, identify the substances labelled A and B. So first off, let's have a look and see what we've got and what we're given. So we know glucose is being made at the top. Now we know G3P is being made in this direction from left to right. And we know carbon dioxide is going to be a substrate in this reaction. So when it says identify A and B, well, we know A, it must be ribulose bisphosphate because we know that's fixed with carbon dioxide by the activity of the enzyme, the catalytic activity of Rubisco. Now that forms the unstable 6-carbon, but we know that splits into 2 times G3P in the stroma. And then it says what's B? Well, we know B must be triose phosphate because we know that's made from the reduction reaction involving ATP and reduced NADP from G3P, glycerate 3-phosphate. Now it says what type of reaction is taking place at number 1? Well, it must be carbon dioxide fixation. through the catalytic activity of rubisco. And then it says, describe how the two products from the light-dependent reaction are used in this stage of photosynthesis. So we could actually draw these onto the the diagram here. So we know ATP is going to be needed, and that's going to form ADP and inorganic phosphate. That hydrolysis provides 30.6 kilojoules per mole of energy, and that energy drives the reduction of G3P to triose phosphate, regenerating the oxidized NAD